We are going to have a presentation from Andy Karran, who is the evidence officer for the Gwent Wildlife Trust. And he is speaking about the Trust's recent survey work using eDNA. So this should be a very interesting talk. So without further ado, I'll hand you over to Andy.
Thank you, Alison.
Sorry.
Same problem with things in my way here. There we go. Right, yes, I'm going to chat about eDNA. I don't claim to be any kind of expert on this, but at the Trust, we were lucky enough to get some funding this year to do some eDNA work. So we did. So first of all, what is eDNA? It's environmental DNA. So it's a genetic material that's obtained from environmental samples such as soil, water, air, sediment, and so on, and possibly for the remains of the animals, maybe hair droppings or dead bodies. So it's likely to be an increasingly important tool in undertaking ecological survey work in future years. And certainly at the Trust, we sort of recognise we have charity, we've only got sort of limited money and resources. We need to get sort of maximum value out of the work we do. So that's why we've sort of explored eDNA this year to see if we can get sort of more results back for less money and effort. So a lot of you may well know it, it's sort of well established for doing great crested newt survey work, sort of presence like the absence, a good substitute for torchlight and bottle trapping surveys, which can be sort of quite labour intensive and involve being out at night time. And it also used maybe if you collect bat droppings from roosts, you can send them off, and with the eDNA you can find out what bats are in the roost. But we sort of got money this year to do work on dung beetles, invertebrates in the reens on the Gwent levels, and grassland fungi, and as a byproduct of that also the grassland flora. So why do we want to use sort of eDNA? So it's good if you've got a difficult to identify sort of species groups. So some people might think it's easy to do grassland fungi and invertebrates and reens, but they are quite challenging and sort of need an expert really. So if you can use the eDNA, you can maybe get some survey work done by people who maybe aren't experts but can go out and collect the samples. And the grassland fungi, as well as being quite difficult to identify, they're quite unpredictable when the fruiting bodies will appear, and very sort of ephemeral. So they may just appear for a couple of weeks, late autumn. But with the eDNA, you can do the survey work all year round. So these are sort of great candidates for doing eDNA work. So first thing we were going to do was dung beetles. And we got this methodology from some work that had been done in Spain, where we were going to put in pitfall traps, bait them with dung, and the dung beetles were going to fall in, and then they were going to shed some of their eDNA down into a solution in the bottom, which we're going to collect. And we were just about to go ahead with this, and then we were told we really need to kill all the dung beetles. And I understand doing sort of recording, sometimes you do need to kill them, but as we had a methodology that didn't involve killing them, then we were told we had to, we decided not to. So we shelved that, and we quickly got a new sort of project, and this was eDNA in the reens on the Gwent levels. So you're probably all familiar with the Gwent levels, a huge network of kilometre after kilometre of reens, ditches. Virtually all of it is SSSI, and the main reason it is SSSI is because of all the life that lives in the reens. And but you really do need a real expert to be able to identify what's in the reens. And yeah, so it's quite tricky work. So we thought if we can just collect water samples from the reens, send them off to an eDNA company, we can maybe get some good results back. So we secured a bit of money and did a bit of a pilot study. So we just did 10 different reens, but we looked at ones of different shading, sizes, orientation, and sort of management. So we can see if we got different results from how different reens are managed and sort of surrounding habitats it was relatively easy to undertake the sampling. We had to be very, very careful to avoid no contamination. So basically involved ladles, syringes and filters. And we ended up just with like a filter paper, which had loads of water pushed through a syringe, which we then sent off. But there was lots of single-use plastic, which wasn't ideal, but that's what we needed to do. So we sent off our 10 samples. 2 of them failed. I'm not quite sure why, but we didn't get any results back from them. But we did from the other 8. So we got, I can't see at the top of my page, I think we had about 180 different species returned. So we got average of taxonomic richness between 13 and 86 species per sort of sample. And we got lots of quite obscure things in there. So there was little phantom midges and non-biting midges and a small square gilled mayfly species, was some of the commonest. But most interestingly, got all the records back, just a huge, big, long list. But looking at confirming against ADERIN records and NBN records, there was 7 which had apparently never been recorded in the UK. 17 species were, including the ones that hadn't been recorded in the UK, were new to Wales. 25 that hadn't been recorded in Gwent, and there was a number of species on the IUCN red list as well. So it's probably going to need further work to find out whether these are actually new records for Wales and the UK, or possibly they have been recorded but not sort of properly recorded. You know, people, it's just their records are sat at home and haven't been entered, but it sort of showed the possibilities of doing this work. There's all these obscure species there, some of which may be very rare, which people hadn't surveyed before, but we managed to find them. There were obviously some limitations. We sort of found hard-bodied invertebrates such as beetles are likely to shed less eDNA into the water, therefore they're likely to be under-recorded. There's certainly be some species that they haven't been sequenced yet, so they're not on the eDNA database, therefore we're not going to get results back from them. We had to collect our water from mid-water, therefore there may be more eDNA in the silt at the bottom, so we didn't get that. But we feel if we did a bigger data set, and I think that eDNA technology is always progressing, so I think it was a good pilot study, and it certainly got use, but I'd say it probably needs a bit more work, but hopefully in the future it could be a very valuable way to sort of monitor the health of the Gwent levels. And we did a report on this. My colleague Larry put that together. That is available on our website if anybody wants to look at all the finer details of it all. And the other main thing we did was grassland fungi. This is probably a more well-established sort of eDNA. Quite a bit of work has been done over it over the last probably, I don't know, 7 or 8 years. Aberystwyth University leading the way on this. So we sort of teamed up with Aberystwyth University. Here's a nice selection of all the different grassland fungi there. It's I should say, you do the eDNA work. I went out and did all this in April and May, and I didn't see a single fungi. So it's a good technique, but nothing beats actually going out and seeing all this beautiful fungi, all the different colours and shapes. But, so what are the grassland fungi? We're basically looking for the Chegda species. That CHEGD stands for the sort of first letter of the 5 key fungi groups. So there's clavarioids, which are your spindles, clubs, and coral fungi. The Hygrocybe, which is your wax caps, your entelomas, your pink gills, your geoglossum, which is your earth tongues, and your dermaloma, which are your crazed caps and their relatives. So we surveyed 30 sites across Monmouthshire and Newport, and all the sites we surveyed were considered to be suitable for grassland fungi. Some were well-known sites, already well surveyed, so we had species lists from the fruiting bodies. Some were well-known sites but had been sort of poorly reported. Some had just had like maybe 1 record of a fungi there, and some sites looked suitable, but there was no fungi recorded at all. So we went out, and it was basically, you might have a whole field, but you can only cover a 30 by 30 meter area. So we picked the most suitable part of the field, and we took a core sample from the middle of the 36 5 by 5 squares that you get within the 30 by 30, and collected the soil, put it in a bag, sent it off to Aberystwyth University. And then we got our results back, So over the 30 sites, we had 100 different of the Chegda species, 28 sort of spindles, 30 different waxcap species, 26 entelomas, 14 the earth tongues, geoglossoms, and 2 of the dermalomas. So we found a diversity range from 13 to 52 species, and where we were comparing sites where we already had good records from, we found the waxcap numbers were about the same, but the eDNA returned far greater diversity of, sort of clubs, entelomas, geoglossoms, and dermalomas. So they're more difficult to identify, and rather than the really showy waxcaps, they're sort of smaller, more obscure, but the eDNA sort of pulled them all out. So even for very well surveyed sites, we found new species. And there was one site which I thought looked like a really good field. The owner assured me he'd never seen a single piece of fungi in his field ever. But for eDNA, we found 40 different species, including 14 waxcaps. Of particular significance was we found contrast between 2 immediately adjacent sites. This was like Sambos and K published a bit of this in the latest British Wildlife, but it was one side of a fence. There was amazing waxcap fungi. The other side of the fence had been ploughed 40 years ago. And even though these 2 fields were exactly side by side, even after 40 years, we sort of demonstrated that hardly any grassland fungi had moved back in, which really demonstrates how important it is to preserve these sites now, because if you ever lose them, they're really incredibly difficult to get back. And there's a bit of a byproduct from the fungi. We also got all flora records back as well from these 30 sites. So 100 species, 77 species of herb, 23 species of grasses and sedges. And for all these sites, we'd already surveyed conventionally for flora, particularly you can indicate the species of their quality. So we found using the eDNA, we got between 25% of what we got with traditional results up to over 100%. We got even more records than we did with traditional methods. On an average, the eDNA returned 62% of what you get with the traditional method, which was pretty good considering when we surveyed a site, normally we'd survey the whole field, and this just involved a 30 metre by 30 metre square. So this was all the sites, whether we considered them local wildlife site quality from our traditional methods, and whether using eDNA could also help to flag up whether they were local wildlife site quality. So you can see there was a pretty good correlation. For some of the sites, we didn't get quite enough species with eDNA, but even then it would demonstrate that the site certainly had quality, so it would be worth surveying. So if you could just send volunteers out to collect soil samples, and if they came back showing it was good species rich, then maybe the botanical expert could go out and survey in more detail. So the flora is generally pretty good, but there were some things the eDNA couldn't really split up. So there's things like creeping and bulbous buttercup. It was one or the other. And I know in all like 30 fields, there was lots of field wood rush, but for whatever reason, eDNA only came back with like it being present at 7 of the sites. So there's limitations to it for the flora, certainly. And we had crested cowbeat came back from one of the sites, which I think would probably be a new record for Wales, but we're pretty sure that isn't there. So there's the odd little sort of glitch in it. So conclusions. Good news is, because you're all sort of recorders and you probably like going out doing stuff by their traditional survey methods, it's never going to replace us. However, in the right circumstances, it's very sort of effective. And it's one of the limitations is it'll detect if something's present, but it gives you no idea of abundance or where indeed that is recorded within that 30 metre square. I'd say the aquatic invertebrates needs more work. Grassland fungi, I think, was a very useful. I think that's been proved in the past, and we sort of proved it again. It's really, really useful because, as I say, if you're doing a normal survey, you'd probably have to go out 2 or 3 week period, sort of end of October, early November, whereas you could survey this all year round. And the grassland flora had limitations with the abundance and distribution, but it was a useful tool. So, and just a quick little plug here, I'm sure a lot of you live in Gwent, and I'm sure you probably, a lot of you are Gwent Wildlife Trust members, but if you're not and you would like to join, then please do, because it helps us do all our work. So thank you very much.
Thank you, Andy. Fascinating bit of work, actually. Great to see that you've done a lot of work on the Gwent levels. Historically, I was involved in designating that whole area as a site of special
Right.
in my former life. Yeah, fascinating talk. We've got a few questions that have come up on the Q&A. The first is from Paul Seligman, and he says that because the eDNA is detecting so many rare species, it may suggest that these species are under-recorded in the past, which raises the question that if eDNA becomes widely used and these species are found more commonly, we can't conclude that they've increased in number or even stayed the same, as the hugely improved recording approach may mask the real trends in abundance. So it's something that probably need to be aware of.
Yes, certainly. There was, I can't remember the name of one of the fungi, but it's I think it was one of the pink gills, which historically has been known to be very rare, but in our surveys and in other surveys, it's been found to be far, far commoner, and it may be one that probably fruits very irregularly, so it doesn't get picked up in fruiting body surveys, but it is actually known to be present in a lot of grasslands. And also using this Chegda scale, and then how many of the species, you get a certain score, and that says whether your site's locally, sort of nationally, internationally important. But if there's more eDNA work, you're going to get higher numbers of records. So the whole assessing whether something's locally, nationally, internationally important will probably need to be re-looked at again as well. So yeah, quite possibly it's going to change things a bit, but it's, I guess the more records you get in, the better. So it's all good.
Excellent. Thanks, Andy. We've also had a question from Soren, and it was something actually that I had also written down myself. And that's about the persistence of eDNA in the environment. Could we be recording species that are already functionally extinct? So how long does that eDNA stay viable? Has there been any research done on that?
I think there has, and I said I'm no expert on all this, but certainly with the fungi, I felt we were actually collecting sort of almost live mycorrhiza hyphae in the ground, because I was told I had to get the soil home, get it in my fridge, get it sent off by DHL the next day. It denatures so quickly and you lose your records. So I feel, certainly with the grassland fungi, I think if we were recording it, it's there and it's living in the field. But there's certainly other stuff I think could be persisting for longer. So yeah, I think there's research going on into this, because you don't want to be picking up eDNA and saying it would still be of interest, but if the thing had been dead for 10 years and then you suddenly think, oh, this is here, we're going to do all the stuff to conserve it, and the species is actually gone.
Yeah, well, that's great. Thanks, Andy. I mean, it is a fascinating subject, because it's clearly a new way of recording. So it goes against the grain a bit, a sort of hard line, getting out and actually seeing the things in the flesh. But certainly as a tool to sort of get an idea of what's in the environment before you start would certainly be very helpful.
And there's certainly no substitute for going out and actually seeing these things, because as I said, I didn't see any fungi, and I was just collecting water out of a ditch. So it'd be far nicer to have seen all the living creatures. But yeah, it was useful. I think we will be using it again in the future. That's
great. I think Dave has asked, given that the eDNA is shown to produce some erroneous species like the crested cowwheat, for example, should we not try and find these very rare invertebrates in real life? That's obviously relating to the Gwent levels work that you were doing, because it sounded
it would be interesting if somebody followed up and sort of went back to where we took the surveys and actually tried to find these and definitely prove they were there. I wonder if some of them could even be even to identify these tiny sort of little midges and be sure of it, kind of thing. But yeah, it certainly would be, to have studies where there was traditional and eDNA surveys going on almost side by side would be sort of fascinating. We were going to do that with some invertebrates in Piercefield Woods near Chepstow, but we were going to collect the sample, send half of it off to for eDNA, and half of it was going to go to an expert who was going to identify all the little fragments in our sort of antifreeze, whatever we were capturing them in. But that's something for the future.
Yeah, excellent. Oh well, it's a great piece of work to have done, and it sort of raises the profile of that type of identification, and and I think it will be a very useful tool to have in the future. Yeah. So thank you very much, Andy, for that.
Thank you.